Rob Schott. I'm here from Sangamo Therapeutics to talk about beyond gene therapy. I hate to compete with lunch, so I'll try to be entertaining about this. My role is head of development at Sangamo. By trade, I'm a cardiologist. And coming to Philadelphia, I'm reminded when I was here as a student in 1978, when Frank Rizzo was the mayor. 1978, you probably don't recall, it wasn't a good year for Philadelphia. Remembering back to the things that I was studying back then, it included cardiovascular physiology, and thinking about what was available for therapeutics back then, and where the state of medicine was back then, and comparing it to now it reveals a breathtaking journey of progress. Back then, we were lucky to recognize rare diseases, and now we can actually begin to think about new approaches, and we have quite a few approaches to treating rare diseases. My father died the following year of heart disease, 1979, and this was even before statins were available. You did have surgical approaches to heart disease with bypasses and things like that, but all of this was absolutely on the horizon. Gene therapy and even biologic therapy with antibodies was not available at that time. We only had small molecules for therapeutics. So in the course of my career, I've seen therapeutics advance from occasional recognition of rare diseases based on clinical features through to the molecular diagnosis, through the development of biologics, the development of gene therapy. And what I'm going to talk about today is what's beyond gene therapy and what we're doing to advance treatment of rare diseases in that space. So we're going to talk about genomic medicine approaches, including cell therapy, Tregs and CAR Ts. I'll explain those a little bit as we go along here, and then what we're doing with genome regulation. So we'll start with cell therapy. And Sangamo uses a gene editing suite of tools called zinc fingers. There are a number of suites a number of toolboxes that can be used to edit genes, and these include the CRISPR-Cas9. I think that probably occupies most of the mind space here. But before that, there were zinc fingers. There's also TALEN. So you have a number of editing tools that will allow you to make cuts or alterations in the DNA. So the zinc finger platform can be used to create genomic medicines for ex vivo cell therapy. So ex vivo cell therapy involves taking cells from the patient, either stem cells or effector T cells or regulatory T cells, and then modifying them to target diseases. In the case of stem cells, we can target hemoglobinopathies, including sickle cell disease and thalassemia. Effector T cells are modified to treat cancer, and then the regulatory T cells are used in transplantation and autoimmune diseases. Now, I want to introduce the T cell to you just briefly. T cells are derived from the thymus. That's where the T comes from. And we have a complement of these at birth, and then they dwindle throughout the rest of your life. And your T cell population begins to collapse as we get older. And there are two general important populations within T cells. There are the T effector cells, which are characterized by the presence of a protein on the surface called CD8, more colloquially known as killer T cells. So these are the Marines that go and blow stuff up. So when you have a bacterial infection or anything that's recognized as foreign by the immune system, the CD8 cells, the killer cells, go in and release their magic, their cytokines, and kill everything in sight. They're like Marines. Go in and kill everything and let God sort it out. And then you have the CD4 cells that are referred to as the helper T cells. And they provide lots of assist functions for the immune system to call in the signaling core, in part, to call in those killer T cells. There's a subset of those helper T cells that are the regulatory T cells. About 1% to 4% of the circulating helper T cells are T regulatory cells, and they're like the fire department. They're going to shut everything down. So we'll keep that in mind. We've got killer T cells, we've got helper T cells, and then we have the fire department regulatory T cells. And these are things that we can go in with zinc fingers to modify the genes in these cells, reinfuse them into patients, and address diseases this way. So the first disease I want to talk about is sickle cell disease. This is characterized by intensely painful sickle cell crises. This is what, when I was a resident and a doctor early in my career, we called them sickle cell crises. Now they're referred to as vaso-occlusive crises. And it's like putting a belt around your extremities or around an organ and cutting off the blood flow, because these sickle cells are ineffective at delivering oxygen to the tissues. The disease is also characterized by anemia and susceptibility to infection. And you have a single nucleotide polymorphism on chromosome 11 that gives rise to a single amino acid change. If you have one of these chromosomes that you inherit, one of these alleles that you inherit, you have sickle cell trait. And this protects you against malaria. This is why this trait has been conserved, because it protects you against malaria. Interestingly enough, elite sprinters at the Olympic level, they have a high prevalence of sickle cell trait. It may assist in explosive exercise performance. So normally, as you can see on that panel there, you have discoid-shaped red cells. Sickle cells, because of an abnormal sickle hemoglobin, they form long polymers and spikes, and they poke out through the surface of the red blood cell and give it a spiky appearance. And these cells sickle under certain stresses in the blood cells, and they sludge and block capillaries and impede blood flow. So that's what causes these intensely painful sickle cell crises. That is what patients experience. And these are life-changing events when you experience these repeatedly. So the zinc finger approach to treating sickle cell disease is it's an autologous approach. So this is unique for each patient. You extract the stem cells, the precursor stem cells, by the presence of this protein on the surface called CD34, and they use a zinc finger nuclease to knock out the expression of a repressor for a form of hemoglobin, which is fetal hemoglobin. Now, let me back up a second. The first year of life, if you have sickle cell disease, you don't have sickle cell crises, because in that first year of life, you're still expressing a fetal form of hemoglobin, which was resistant to sickling. And then this repressor, this off switch, turns off the production of fetal hemoglobin after the first few months of life. Our strategy is to knock out the off switch. So the off switch is disabled, and fetal hemoglobin production is turned back on. And this is remarkably effective in preventing sickle cell crises. So these engineered blood cells are then reinfused back into the patient. They're stem cells, so they go back into the bone marrow, take up residence, and begin to produce populations of red cells that are equipped with fetal hemoglobin and resistant to sickling. So let's talk about the T regulatory cells and CAR Tregs. And CAR Ts are also something that is fascinating, that got me into this business of gene therapy from the small molecule and biologic land where I existed before. And CAR Ts are chimeric antigen receptors. This is basically re-engineering the surface proteins on the TReg cells so that they express proteins that are like postage stamps, that will take these Tregs and direct them to certain locations in the body where the disease is manifesting. So this is a busy slide. Tregs, again, are a subset of those immune cells, the thymus-derived cells with potent immunomodulatory functions. Tregs, as we talked about, terminate inflammatory signaling, the fire department subset of T cells. They go in and tell the Marines, the killer T cells, to knock it off. And by preventing excessive or inappropriate inflammatory responses, the Tregs help to prevent autoimmune inflammation. Autoimmune disease is recognizing self as a target for the most part, and you don't want to recognize yourself as a target. Too little recognition, and you can have cancer. It's the yin and yang of diseases, cancer and autoimmune disease. Cancers evade detection by the immune system. Autoimmune diseases, there's too much recognition. Of course, genetic deficiencies in Treg cells can result in lethal autoimmune syndromes, the bubble boys, and the SCID, the severe combined immunodeficiency is a defective function in these populations of cells. T regulatory deficiency or dysfunction is described in multiple autoimmune diseases. And these T regulatory functions, in addition to turning off immune signaling, help to maintain immune tolerance and promote tissue regeneration. So in the panel here, it shows normal T cell function. You have the T effector cells attacking and invading bacteria here, and the TReg cells here, shown in red, help to turn off the inflammatory response, the inappropriate response. So what's the promise of Treg cell therapies? We would like to potentially see this as a first-line therapy for autoimmune diseases or other forms of disease. Currently, we have anti-inflammatory medications that we use. The one you probably all recognize are systemic corticosteroids, which are associated with a tremendous number of side effects because they work systemically across all immune functions to shut off the immune system. And they can be associated with diabetes and weight gain and a host of unwanted side effects. And you have to take these continuously and take them in bursts when inflammation is more turned on. The hope is that we'll have T cell therapies which leverages the entire array of cellular anti-inflammatory signaling. These Tregs are equipped with cytokines multiple cytokines are basically a factory that produce signaling proteins that will turn off the inflammation. And they can interrupt these pro-inflammatory feedback loops and modify the typical course of the disease. And you can target them to very specific areas of location by modifying the T cell receptor, these so-called CAR-Ts. And they could be of durable efficacy by leveraging the body's natural mechanisms to switch off inflammation. So this is the first application of Treg therapy in patients. We dosed our first patient with this therapy this year. And what we're doing is we're trying to prevent kidney transplant rejection. And what this slide tells you about is a mismatch in these patients in what's called a minor leukocyte antigen. HLA is human leukocyte antigen. So when you get a transplant, you're matched for the major histocompatibility antigens, MHC1 and MHC2. You have to have a match. But there are minor antigens with a living donor that you can't match for. There's more than 30 different antigens on the surface, and you only match for the major ones. You don't match for the minor ones. And there are about 25% of patients have an HLA-A2 mismatch that can promote rejection of the transplanted kidney. So in our study of this, the Steadfast TX200 study, it's a phase 1-2 trial of genetically modified Tregs engineered with a receptor, a CAR-T, to hone Tregs on the kidneys that are transplanted with an antigen mismatch. So basically what we do is we create this chimeric antigen, so it's a T cell, part T cell receptor and part A2 receptor. So it's a chimeric protein that, because of this receptor, hones in on the kidney, which has the A2. So the donor has the HLA-A2 antigen, the recipient does not. So the only place for this to go is where the postage stamp mails it right to the organ. That's the only place that you're expressing the HLA-A2 antigen. And the idea there is that it's going to switch off inflammation, which results in the rejection of the transplanted kidney. That's our proof of concept for this. And as far as we know, we're the first company that has dosed a patient with modified Treg cells. Again, the zinc finger technology is being used to engineer these chimeric antigen receptors, the CAR-T cells. This talks about how this is done a little bit. We basically use vectors to introduce the blueprint for those new chimeric antigen receptors. And in the process, we attempt to restore healthy balances between the effector cells and the and the regulatory cells. So we add the Tregs, the helper cells with the Tregs now restores immune balance. With rejection and with autoimmunity and with inflammation, you have a mismatch between the effector T cells and the regulatory T cells. So right now, this is an autologous process, meaning that each patient is their own donor. So we take the patient's white cells, this process called leukapheresis, where they're extracted from the patient. You select out the TReg subset. This is, again, a very small proportion of the circulating white cells. You use a gene transfer technology and zinc fingers to make a cut and insert your new program, your new blueprint for these chimeric antigen receptors, the postage stamps. And then you expand these ex vivo before you put them back in the patient. You freeze them, and then ultimately you infuse them back into patients, usually 8 to 12 weeks after the transplant. They get an infusion of these engineered T regulatory cells that are designed to go right to the kidney and hopefully switch off the anti-rejection inflammation, or the inflammation that is responsible for rejection. So what we're working toward is an allogeneic cell therapy approach. This would greatly reduce the cost burden associated with this type of therapy, because now it's very much tailored to each patient. Each patient is their own donor. With allogeneic cell therapies, you can get a universal donor. This is kind of like the ABO blood type system, where type O is the universal donor. You get a universal donor for the T regulatory cells. You'd engineer them using the zinc fingers to arm them with the appropriate cell surface receptors, the CAR-Ts, and then you can expand them and then store them so that one donor could then potentially provide off-the-shelf engineered cells for lots of different patients and greatly reduce the cost associated with this type of therapy. In order to do this, what we have to do basically is knock out those major histocompatibility antigens, those proteins that make each of those white cells unique to us. Mine's different than yours, and we all have our unique population, but you can knock those out potentially and create a generic type of T regulatory cell. And that is the objective of allogeneic cell therapies. And finally, I'm going to talk a little bit about genome regulation. Gene therapy started, there's been a couple thousand patients dosed with gene therapy. We are replacing the defective gene. In the example I'm going to give of genome regulation, we're going to regulate the genome. That's the objective. And zinc finger transcription factors can be used to regulate gene expression. We're looking in the central nervous system to do this. They can be coupled to transcription factors. So zinc fingers are basically a recognition system. They can interrogate 3 billion base pairs with fingers and then lock onto any specific location, any specific address on the DNA and park there. So zinc fingers allow you to very quickly figure out where it is that you want to sit on the DNA. And then you can attach to those zinc fingers various proteins with functionality, including the regulation of transcription factors. And with these zinc finger transcription factors, you can fine-tune expression based on the genetic needs in each disease through modification of the zinc finger structure. And this can be used to reduce the expression of a pathogenic gene or selectively repress expression of a mutant allele. You get one from mom, one from dad, one's defective. You can suppress expression of the defective one and not touch the healthy allele. And that's based on the high specificity of the zinc fingers to be able to recognize the sequence around the diseased allele. You can also activate the expression of genes that are inadequately expressed. So what we do in order to work this magic is to package these zinc finger transcription factors in a capsid, which in this case is an AAV, an adeno-associated virus. And then you administer this to the patient. Several routes of potential administration. One is systemically, one is in the cisterna magna, the other is in the cerebrospinal fluid. You can also potentially directly inject it into the brain. And then these zinc finger transcription factors find the target gene using the zinc finger addressing mechanism, and they can, in this case, a transcription factor can switch off the expression of the gene and reduce ultimately the production of an unwanted protein. Similarly, you can use a zinc finger transcription factor to increase gene expression to get more production of a desired protein. So we'll start in this slide with the diseases. So neurodegenerative diseases across the board are characterized by the accumulation and misfolding of unwanted proteins, what I've referred to as protein clumpopathies. This is true in Alzheimer's disease, in Parkinson's disease, frontotemporal dementia. Some of the diseases here are listed that are characterized by the accumulation of unwanted proteins. There's the tauopathies like frontotemporal dementia. Also, CTE, chronic traumatic encephalopathy, is associated by the accumulation of this protein called tau. Parkinson's disease, alpha-synuclein is the signature protein that accumulates. Huntington's disease, it's Huntington, is the protein. ALS, there's a couple. C9orf72 is one. SOD1 is another one there that accumulates in the motor neuron cells that cause amyotrophic lateral sclerosis. And then prion disease, it's the prion protein that accumulates. Mad cow disease. So this is what happens in normal physiology. You have DNA expressing with the gene that then information flows to the RNA. You have sense and antisense in RNA. You may have a misspliced RNA, but you end up with RNA that specifies a protein that is abnormal and clumps and accumulates in neurons, is ultimately toxic to the neurons. So all of these different diseases have the same theme, and that's the accumulation of these complex proteins that over the course of time accumulate and are toxic to neurons. Different populations of neurons produce different types of diseases. If it's alpha motor neuron, you get Lou Gehrig's disease or ALS, and if it's more global, you might get a frontotemporal dementia. So what we do with the zinc finger approach is to target these genes and reduce the expression. So you don't get the RNA and you don't get the protein. So you can eliminate, in theory, the accumulation of these proteins which are responsible for the manifestation of these various CNS diseases. Here we talk about genome regulation approach in tauopathies. Again, pathologic, mislocalized tau aggregates and tangles, like frontotemporal dementia. And we can deliver a transcription factor with the zinc finger nucleus. It takes us to a very specific place in the DNA, and we can reduce the production of the tau protein. We can cure this in mice, actually. Same thing with cancer. But in humans, what we want to do is restore healthy neurons by knocking down the expression of the tau proteins across a variety of tau-related diseases. And here we're targeting amyotrophic lateral sclerosis, also known as Lou Gehrig's disease, at the genomic level with allele-specific repression. So we're not going after the healthy allele, where you get the appropriate C9 open reading frame 72 normal isoform of the protein. We're targeting the diseased form of the protein that accumulates and causes the death of the neuron, the motor neuron. And again, what we do is we can administer an AAV with a zinc finger transcription factor that recognizes only the unhealthy allele, not the healthy allele. So you can get normal expression of these proteins. This is the promise of in vivo gene editing and the promise of zinc fingers. So with that, I'll conclude. I know that was a lot of information in a short period of time, and I think we have a couple minutes for questions if you have any questions related to this.
Great talk. I had a question on the CAR TReg therapy in the kidney transplant setting.
Mm-hmm.
Just about the dosing and frequency, you mentioned that the therapy was administered 8 weeks after transplant. Can you comment on whether or not that time frame would be applicable in the auto versus allo setting, meaning that in the allo setting, is that too long? Would you have to go early? And is the idea to wean off traditional immune suppression post-transplant?
Yeah, so the objectives of the study, the outcome measures will be reduction of rejection, but also with time, the reduction of those systemic anti-transplant medications, corticosteroids, the rapamycin-like drugs that are used you have to chronically administer that. So in picking the time that you administer the therapy, it's an educated guess. You don't want to, there's inflammation associated with healing, so you want to allow the kidney to engraft and to begin to heal. But then the rejection occurs over time. So what we're able to do, what we're intending on doing is monitoring biopsy specimens and other biomarkers to look over time whether these CAR Ts are persistent, whether they get in there, because you can see them. You can actually histochemically stain for them and see that they're present in the kidneys. And kidney biopsies are a routine part of post-transplant monitoring. So the first step is to show that these CAR Tregs take up residence in the transplanted kidney, and then over several years, you're going to look, compared to a control group, at how much anti-rejection medication systemically that they require. And the objective here is to, one, overall reduce the number of rejected kidneys, but also reduce that burden of systemic anti-rejection medication.
Just a quick follow-up question. Do you anticipate repeat dosing, or is this envisioned as a one and done?
Excellent question. And we don't know at this point how long these are going to persist. We'd like it to be one and done, and that you have these cells that persist, but it's entirely possible with allogeneic therapies. One of the advantages there is that you potentially could redose if you needed to. This is all very new science, clinical science, and we're still trying to answer these questions.
Thank you.
Thank you.
Rob, nice presentation. First thing I want to say is you did a really great job of explaining such complicated science to those of us in the room who don't always understand it, and a lot of the people who understand it so well don't realize that the gap between their knowledge and the knowledge of the people they're talking to. So I thought you did a particularly good job of helping us to understand some really complicated science you were describing. Can you tell us the difference between zinc finger and some of the other gene therapy modalities that are out there, like what's better or not as good about it, and where is it useful and not useful compared to CRISPR-Cas9 and some other things?
Yeah, so zinc fingers have been around for a long time, and Sangamo's had a lot of the IP around that. So they haven't been used as broadly, in part because we own them. We've licensed them as research tools. This company's been around for 25 years, initially based on zinc fingers and providing these to various labs and research groups. CRISPR-Cas9 is more recently entered the realm of zinc fingering tools. The differences between zinc fingers, some of the differences between zinc fingers and CRISPR-Cas9. CRISPR-Cas9 comes from a bacterial system, where zinc fingers are human-derived gene editing tools. CRISPR-Cas9 tends to be a little bit bigger. It's based on a different mechanism. It uses a guide RNA to take it to the proper address, whereas we use DNA recognition repeats. They're both effective at editing, cutting the DNA. Some of the advantages of zinc fingers is that you can attach all of these transcription factor modulators to them. So you've got your basic zinc finger recognition tool, and then you attach the editing function proteins to that. So we can upregulate, downregulate, cut, double-stranded cut, single-stranded cut with zinc fingers. So typically, companies in the CRISPR-Cas9 space are focused on single types of functionality, whereas we like to think that we have more functionality with respect to what we can do with genetic or epigenetic engineering. But it's all very useful technology. The CRISPR-Cas9 really was described for use in humans around 2012. That was Jennifer Doudna and Emmanuel Charpentier, who won the Nobel Prize for their work in the CRISPR-Cas9 systems. There's also TALENs that are used for this. So they're all useful. They've all been used in humans, and hopefully we're going to find the best application for each of these to really alleviate the burden of disease. That's the objective. All right, thank you for your attention.